Dear students, Assalamu alaikum. This is lecture number 1 of the series of our online lecture of the course Molecular Biology, course code BCHM221. The topic of our lecture today is transcription in eukaryotes. So before going to the course outline, I would like to remind you that we had talked in great detail about the process of transcription in prokaryotes. Talked in great detail about the process of initiation. Then we talked about in a separate lecture about the process of elongation and termination of transcription in prokaryotes. But I think it is necessary to understand the process of transcription in eukaryotes, because this is how it can give us very useful information of the differences in the process, the similarities, and how they can possibly be exploited to understand the therapeutic targets that they can offer, once we have knowledge of these things. So this is why I prefer to talk about the process of transcription in eukaryotes in separate detail. So now the outline, we'll talk about the difference between prokaryotic and eukaryotic transcription. This is very important or fundamental to understand, because these differences, as I told you before, can function or can work as a source of therapeutic target. Now I talk about eukaryotes nuclear RNA polymerases, because they are different from the prokaryotic RNA polymerases, which are present in the cytoplasm, because in prokaryotes we don't have any nucleus. So the eukaryotic nuclear RNA polymerases are the ones that are located in the nucleus. And then we talk about transcription factors. Transcription factors play a very pivotal role in the process of transcription in eukaryotes. We talk about steps in eukaryotic transcription. So this will be also we'll talk about. Now before going into the details, we focus on the first step of the difference between prokaryotic and eukaryotic transcription, and then we move about the detail of the transcription. Now, differences between prokaryotic and eukaryotic transcription. Prokaryotic transcription takes place in cytoplasm. As I told you before, the prokaryotes, they don't have nucleus, so their nuclear material is in the cytoplasm, whereas in eukaryotes we have eukaryotic transcription that takes place in the cell's nucleus, and then messenger RNA comes to cytoplasm, and then the process of translation occur in the cytoplasm. But here in prokaryotes, you can see that the process of transcription is the one that takes place in cytoplasm, and along with that, translation also takes place in cytoplasm. So both the process of transcription and translation takes place in cytoplasm, whereas in eukaryotes the difference is transcription occur in cell's nucleus, whereas messenger RNA is transported to cytoplasm, and then the process of translation occur in the cytoplasm. Now this is not the only difference. There are many other differences, including DNA in prokaryotes is more accessible to RNA polymerase than DNA in eukaryotes. So let's say about accessibility. The accessibility of prokaryotic DNA is much more as compared to that in eukaryotes. Now eukaryotic DNA is wrapped around proteins called histones to form structures called nucleosomes. In eukaryotic, DNA is packed, that is in the form of chromatin, while RNA polymerase interacts directly with prokaryotic DNA. Other proteins mediate the interaction between RNA polymerase and DNA in eukaryotes. Messenger RNA produced as a result of transcription is not modified in prokaryotic cells. Eukaryotic cells modify messenger RNA by RNA splicing, 5' end capping, and addition of a poly A tail. This is how the post-transcriptional modification occurs in the eukaryotic cells. So here is an overview of the difference between prokaryotic and eukaryotic transcription. This is how prokaryotic cells looks like. The DNA is here. Here is, you see, there's no nucleus. The process of messenger RNA, and this is messenger RNA that has formed, and here the process of translation is taking place. Growing amino acid chains are shown here by these round shaped structures. So this is how it happens in prokaryotes. Whereas here, this is the DNA inside the nucleus. You can see the round shaped structure. This is how the process of transcription and processing occurs inside the nucleus. We have in the red, you can see the formation of the RNA. Here is the cytoplasm. Here is that messenger RNA. Now, this messenger RNA is transported out of the nucleus to the cytoplasm, where you can see here the red, and then the translational machinery is helping to synthesize the growing amino acid chain. You can see here about this round structure. These represent amino acids. This is the process of translation. So you see that here process of transcription takes place inside the nucleus. It is processed and then transported out of the nucleus to the cytoplasm, where the process of translation takes place. Where it is compared to that prokaryotic is a simple process. Everything is happening in the cytoplasm. So this difference is important to understand. Now, eukaryotes nuclear RNA polymerases. Eukaryotes have 3 nuclear RNA polymerases, each with distinct roles and properties. Now, RNA polymerase 1, Pol 1 or Pol A, is transcribed in nucleolus. Larger ribosomal RNA or RNA 20S, 28S, 18S, and 5.8S. There's RNA polymerase 2 or Pol 2. It is also called as Pol B. It is transcribed in the nucleus. The product is messenger RNA, mRNA, and most small nuclear RNAs are snRNAs. There is another type which is called RNA polymerase 3 or Pol 3. It's also called as Pol C. It's transcribed in nucleus and possibly the nucleolus nucleoplasm interface. Transfer RNA, tRNA, and other small RNAs, including the small 5S RNA. Now, this is something that we briefly talked about, but we have not talked about in detail, which is transcription factors. Transcription factors play a very important role when it comes to eukaryotic cells. Transcription factors in eukaryotic cells have a very important role in the process of transcription. So they function as activator proteins, bind to pieces of DNA called enhancers. Here you can see the enhancer region. Their binding causes the DNA to bend, bringing them near a gene promoter even though they may be thousands of base pairs away. So here is the activator proteins, the regions of enhancers. Other transcription factors: protein join the activator proteins, forming a protein complex which binds to the gene promoter. Then this protein complex makes it easier for RNA polymerase to attach to the promoter and start transcribing a gene. So this is just a diagram that simplifies the DNA greatly and shows some key regions like enhancers, activator proteins, show some genes, some promoter region. Then you can see methyl groups here. Here's the insulator, the CTCF, the RNA polymerase. So this gives you an overall picture, but this does not have each and every tiny detail that we are going to talk about now, the steps in eukaryotic transcription. So like prokaryotes, we have the processes of initiation, elongation, and termination, but in eukaryotes there are slight details which are more advanced as compared to that of prokaryotes. So they include pre-initiation, initiation, elongation, termination, and then processing of RNA that we call post-transcriptional processing. So these are the steps. Now we talk about them in detail. Now you know about there are a lot of things which are in common between the processes of transcription in prokaryotes and eukaryotes. So I will not talk about the details of those things that we have already discussed in transcription in prokaryotes. I have tried to make this presentation in a way that we focus on the uniqueness of the process in eukaryote, and then we can make somehow the comparison between the prokaryotes and the eukaryotes. So like now, transcription occurs in the nucleus of a cell. DNA contains specific section, each of which plays a different role in the process of transcription. So you can see the different regions. This may be a gene, this may be a promoter region, depending on the situation. We talk about them in detail. Transcription unit. The transcription unit is the area of DNA where the gene or genes that will be transcribed are located. This is important to understand, like we had talked about this before, that transcription, the process of transcription is not a futile process. It's not that something randomly is transcribed. No, it depends on the needs of the cell, the needs of the body. It is a very well planned process. So the transcription unit is the only region that is transcribed. It's not the whole DNA that is going to pass through the process of transcription. So look, so here is something important. The TATA, located approximately 25 to 35 base pair upstream of the transcription unit. The TATA box is a highly conserved sequence that works to help position RNA polymerase II during the initiation of transcription. Remember, this is located somehow 25 to 35 base pair upstream of transcription unit. They play a very important role. And then we talk about the enhancer. Enhancer works to stimulate transcription. They are generally located hundreds of thousands of base pairs upstream from the start site of transcription. They are responsible for helping to position RNA polymerase II at the initiation site for transcription. The general transcription factor known as TF2D appears. You see now we have enhancer, a particular region, then we have a TATA box, then we have the transcription unit. Now TF2D is the largest of the general transcription factors with a mass of approximately 750 kilodalton. It is a multimeric protein. In addition to a single TBP, TF2D also includes 11 TBP associated factors. Included in the TF2D complex is a single Tata-box binding protein with a mass of 38 kilodalton, that is shown in yellow here. TBP, and therefore the TF2D complex, is the first protein that binds to the Tata-box promoter region. TBP, with its folded over saddle-shaped structure, interacts with the minor groove in the DNA. This creates a bend in the DNA strand once the TBP is bound. Here is the TF2A, this is the TF2B. In addition to TF2D, there are other smaller transcription factors that helps to promote and assist the initiation of transcription. TF2A is not absolutely required for the initiation of transcription, but it aids with the binding of TF2D. This is the TBP. TF2A is shown here. TF2B interacts with both the TF2D complex as well as helping to secure the attachment of RNA polymerase 2, pol 2. Here is the RNA polymerase 2, and this is TF2F. This is TF2D. This is RNA polymerase 2. This is TF2F. Here TF2B is shown, and here is TF2A is shown. Before binding to the DNA, TF2F and pol 2 join to form their own complex. The pol 2 complex binds to the start site, but two more general transcription factors are required before the DNA can be separated and used as a template. One of the two factors still needed for transcription to begin is TF2E. TF2E must bind to the pol 2 complex first in order to create a docking site for TF2H. TF2H is the last general transcription factor required to complete the assembly of transcription initiation complex. With TF2H and TF2E in place, energy is required to activate the unwinding of the DNA duplex. The energy required to activate transcription comes from ATP molecules. The energy transfer is accompanied by ATP hydrolysis. This results in the phosphorylation of pol 2 complex in the transformation of ATP into ADP. Once the ATP activates the complex, the helicase activity of TF2H subunits results in the separation of the DNA strands. Unwinding the DNA allows messenger RNA elongation to begin. Once transcription has begun and mRNA elongation is underway, all of the general transcription factors except TBP and TF2D are released. TF2E, after being released with the rest of the general transcription factors shown here, elongation of the messenger RNA continues until the entire transcription unit has been transcribed. So once the end of the transcription unit has been reached, the rest of the complex is separated to dissociate. TF2A, or also after being released with the rest of the general transcription factor, TF2D and TBP are still bound to the Tata box. mRNA prepares to be released from the pol 2 complex. The DNA eye closes after elongation ends. So as the pol 2 complex moves off, the phosphates are also released. The messenger RNA is released as the pol 2 complex leaves the DNA strand. The pol 2 complex dissociates, leaving only the TBP TF2D complex attached. So the messenger RNA and pol 2 complexes have both dissociated. Here you can see the last complex to release is TF2D by way of TBP. Once this final transcription factor separates from the DNA, the bend formed by it reverts to its normal shape. The messenger RNA is now ready to be both processed and moved out of the nucleus. Once outside, it can then be translated. The next step in gene expression, the DNA has returned to its original shape. So this is the end of the process of transcription in eukaryotes. I hope you enjoyed the lecture, and if you have any question, please feel free to contact me. I will be happy to answer any of your question. Thank you very much for watching. See you in the next lecture.